Hello everybody, today we want to expose about the obtaining carotenoids with the microalgae Sedemus incrassatus and obtain the characteristics of the astaxine from the microalgae Hematococcus pluvialis. The team was conformed by Matías Ochoa, Leslie Aremi García Valenzuela, and Sandy Guillén Gallardo. We hope you like it. Now we have the carotenoids. What are the carotenoids? The carotenoids are fat-soluble terpenoid pigments composed of 8 isoprene units. They have a linear basic structure with conjugated double bonds that represents the chromosphere group, which absorbs light energy in the visible region of the spectrum and gives the molecule its distinctive coloration. The maximum wavelength at which each carotenoid absorbs depends among the other factors. One of these is the number of conjugated double bonds and the existence of closed rings in the molecule, in such a way that 7 double bonds are required conjugated so that a carotenoid shows a perceptive color. Carotenoids absorb light in the blue and green region of the spectrum and reflect wavelengths in the yellow, orange, and red regions, and repeat after that. The classification. According to their chemical composition, carotenoids are divided in two types: carotenoids or hydrocarbon compounds, xanthophylls or oxycarotenoids, which generally present oxygen in thermal rings or their structure. Another classification or criterion involves the way is the which carotenoids are synthesized in organisms, and according to this, primary carotenoids are those synthesized under normal conditions and that favor ground, while secondary carotenoids are produced under special conditions of stress, mainly nutritional stress such as nitrogen deficiency. Now the chemical characteristic. Their nature or carotenoids are insoluble in water and soluble in non-polar solvents. Its degree of solubility will depend on the subsequent functional groups of their molecule. Carotenoids are very soluble in petroleum, aid, and hexane, while xanthophylls dissolve better in methanol or ethanol. Then, the solubility of carotenoids is a very important characteristic sign. It directly influences in the extraction and the purification in industrial processes. Other properties of carotenoids are sensitive to the light, to the oxygen, acid, and compounds with high temperatures. It measures degrade or oxidation reactions, and these properties are reflected in other characteristics such as the attenuation of the energy level on quenching of the oxygen singles or blocking of the reactions mediating from the free radiation, the radicals, also called scavenging or tramping. Carotenoids in general are more stable in systems with a high degree of unsaturation, such as photosystems, since these types of systems more easily accept oxygen and free radicals before carotenoids. On the contrary, in systems with saturated lipids, carotenoids shows greater instability. Consequently, these compounds can act as pro-oxidants or antioxidants depending on the system where they are found. The importance in the human health. Some carotenoids, in addition to giving an attractive color to the different photosynthetic organisms, have the property of presenting activity as a vitamin A, which increases their importance at the psychological and nutritional level. Vitamin A is essential for night vision and necessary to maintain healthy skin and superficial tissues. It can be supplied as such the retinol as some less activity analogous of these precursors, the carotenoids. Not all carotenoids are precursors of the vitamin A, so both provitamin and not provitamin carotenoids are found. The number of the vitamin A precursors carotenoid ranges is between the 15 and 16 high-link the carotenoids and some Xanthophylls have the ability of act as a provitamin A and depends on other conversion to retinol by animals, as well as the presence of ionic carotenoids containing at least one ion 1 ring can be converted to retinol. From this point of view, the most important carotenoid is the carotene, which contains two ionic rings. Currently, the term provitamin A is used for the carotenoids that qualitatively exhibit carotene activity. As for this, its activity will depend in turn of the amount in which is present in food. The state of the amount of the carotene equivale that 1 micro of 4 micrograms, depending of the amount of the carotene present in the food. The importance in the industry of the carotenoids are the health effects to the industrial generation of products such as vitamin and supplements. However, carotenoids have other applications in this area. One is the most important being in function of the pigments in the food industry, since it is considered that there is safe in the consumption of the humans. In poultry farming, lutein is a very important supplement, since the coloration of the poultry meat is one of the quality parameters and also an attraction to the global consumers of lutein is approximately like 300 miles of kilograms per year, and Mexico consumes a lot of this. Same, Mexican poultry consumes more than 15% of the natural lutein basis of pigments. In aquaculture, astaxanthin, mainly of the cataxin secondly, are used as the food supplements for animals such as salmon and crustaceans, which are not capable of the synthesize these pigments and normally absorb them from their marine environment, where they are synthesized by the phytoplankton. When these organisms are ready to be made of the insulated ponds, and it is necessary to supplement them with said pigments in the diet. The advantage of the production of the astaxanthin from the microalgae or many other living organisms is that the subsequent purification is not necessary, since it can be assimilated as it be fish or crustaceans in the culture. These excesses of astaxanthin are a supplement will result in pale meat that is unexpectedly to be the eye. The high price of the pigments make it one of the inputs of the most affected production costs of the salmon industry, representing between the 18 and 22%. Microalgae and carotenoids. The microalgae are microscopic eukaryotic organisms that contain chlorophyll and carry out of the organic photosynthesis. Algae and especially microalgae synthesize a wide variety of the carotenoids, including those synthesized by plants, such as the xanthophylls, biolactin, neoxtalin, and lutein. Carotenoid biosynthesis. Originally, the microalgae carotenoid biosynthesis pathway was believed to be that of the acetate mevalonate. However, lately the pyruvate glycerin hide phosphate pathway has been established for the green algae. And the two routes are they showing in the image. The pathway from mevalonate to prenyl phosphates. The mevalonate is formed from acetyl-CoA and leads to the formation of isopenylene pyrophosphate, which is the temperature forming of the unit of these carotenoids and the microalgae composition. Now we can see the deoxycellulose phosphate pathway to prenyl pyrophosphates and how we can obtain this mediantly the sequence of the structural pathway. Next, we have the carotenoid biosynthesis in the microalgae and how it is carried out of the described above up to lycopene. Subsequently, the cells follow the route described in the next figure. And we can see the lycopene and how it grow up to be manipulated. In relation to the carotenoid biosynthesis, it had several advantages, considering that there are more than 600 carotenoids in the nature. A great biosynthetic capacity can be found in it, without having to develop complex processes of chemical synthesis. Unlike this, by knowledge the biosynthesis pathway of the carotenoid, is it possible to have knowledge for the biosynthesis of several carotenoids. Similarly, the products of these pigments by living organisms prevents the formation of stereoisomers that are harmful to the health. Finally, the biosynthetic capacity is overproducing organisms can be improved to thanks the recombinant DNA, technological and other molecular biology tools. However, it is prudent to also mention the disadvantage of the carotenoid biosynthesis. Biological systems usually produce a mixture of carotenoids which make it essential to apply purification and extraction process, which increases production cost, and another drawback is that these pigments are intracellular components and therefore are not secreted into the medium in the fermentation processes, making their extraction from the cells essentially and consequently production of the cost increase, and this too bad to the industrial and the making other type of cost. In the industrial production of the carotenoids, the chemical synthesis versus the biosynthesis. Well, among the advantages from the chemical synthesis is the purity of the compounds obtained. To their consequently and low production costs stand out of this other hat. It also has certainly disadvantages. The complexing of the synthesis of some carotenoids, although similar processes can be used to produce different carotenoids. Normally, the synthesis of new carotenoid requires the development of all new chemical pathway. Another major drawback of the chemical synthesis of carotenoids is the production of mixtures and stereoisomers, and some of which are not found in nature and are not as activity as those present in it, which is the why they are disabled from consumption due to their unstable side effects. In relation to the carotenoid biosynthesis, it had several advantages considering there are more than 600 carotenoids in the nature, and the great biosynthetic capacity can be found in with having to develop complex processes, chemical synthesis. Unlike this, we knowing the biosynthesis pathway of carotenoids is a possibility to have knowledge of the biosynthesis of several carotenoids in the area. Similarly, the production of these pigments by living organisms prevents the formation of stereoisomers that are harmful to health. Now we have the obtaining and the characteristics as taxing from the microalgae Hematococcus pluvalis. The characteristics are the solvent extraction, the lytic enzyme extraction, astaxanthin extraction be solid and supercritical fluid extraction in the area. And these characteristics have some helpful and produces of industrial uses with the microalgae Hematococcus pluvalis. Some important biotechnological processes for the characterization are the UV spectrum, infrared spectrum, chromatography, and we can have a lot of uses to the biotechnological processes from this characterization. In conclusion, the importance of the pigments is the photosynthesis, in that it helps absorbs energy from the light, and the free electrons are the molecular level in the chemical structure of these photosynthetical pigments, rotate and certainly energy levels. Now we can see the bibliographic references, and it's all of we part.